He's here.
Ma first. Okay, I will bring the people from Teams here and then I'll be back.
Mhm. Hi, Hank.
Hi, Dimitri. Hi,
Hi.
everyone.
How's it going?
Yes, all right. End of the day for me, so I'm just
It's 9 p.m., right?
it's 10 p.m. now because we're on
10.
daylight saving. Yeah, I just had dinner, so I'm full.
That's why you look happy.
So I see Mimi also joined in here. So then all the people from Teams moved to this one. So we're okay again.
So we're gonna be on Zoom, right? That's what we decided to do. For the
Yes,
upcoming
but
weeks.
because the invite automatically links to Teams, there was a little bit confusion with some. Because if you have the Teams, then you can just click on the Teams link and then you will go automatically in Teams. But it's first the Sydney link, then your link, then Teams.
So complicated.
Sorry for that. So
I was still waiting for someone.
Well, Matt said he was on holiday.
Yes, Matt is on holiday, so his apologies, but he won't be in today. But he'll catch up like anyway with the recording, I guess.
Yeah, and well, from my team, we are all online. So how? Do you expect anyone from your team?
It would just be made for today, thanks.
I see that Bernard and Anthony are also there. And Mimi is here. So I think this is it for today. And I noted on the list that I'm not convening. Someone else is, and someone else is also taking the minutes. So
it's I'm leading the meeting today, and the University of Sydney is taking. He's a minute taker.
Yeah, I'll take him. I'm gonna be taking minutes. So.
Yeah.
Okay, then
So
I'll leave it up to
start
you.
off then. Yeah. I don't know. Should I share the screens for the list of things that we should cover, or should I go through them like one by one without sharing the screen? Which one would you prefer?
I think you can just talk as normal, and then when anyone needs an update, they can share the screen. I think.
Yeah, sure. I'm just looking at the GitHub meeting issue agenda. So the first thing that is on the list is biological evaluation, Erasmus in brackets. So, okay, but this is the objective list that had to be done like before the meeting. So the first objective is for Ma to post the PowerPoint presentation from the meeting, from the last meeting in August, which she did like shortly the same day back in August, back in September. Then the next one, so I guess like that's sorted, that's the information we were asking. The next one refers to my, sorry?
Hello.
Hi.
Hi. Oh, yeah. Yeah, we have a presentation. So would you mind sharing my screen?
Yeah, Ma, we were just talking about the previous presentation that it should have been on GitHub, which it is.
Yeah, everything is sorted like with regards to that, so that's fine.
Yes. I uploaded the presentation last month after meeting. So I think the presentation is the slides is in the GitHub now.
I'll skip the second one. I'll come back about it in a second. So the third one for biological evaluation is about the master list. So I can tell straight away that it's up to date now. As you know, like I added like benzoxazoles, all the molecular weights, myos codes, everything. Log D values have been generated by Ben. So this objective is fully accomplished, with exception to the new entries that are being added all the time. So I saw Bernard adding a few metal complexes, I think, and a lot of different like other compounds as well. Yeah.
I don't know. I don't think log D can be calculated for metallo drugs. Can it? I don't think so.
No, I meant like the previous entries from the that were added.
Oh, the previous entries, yes.
Yeah.
The metallo drugs.
Essentially, it's up to date now.
Yeah. Do you want to, Bernard? Do you want to explain a little more about the metallo drugs, or?
Well, I sent Wendy last week some more metallo drugs like oranofin, which is a coat compound, a well-known gold drug used for the treatment of arthritis, inflammation diseases, and it also showed various activities against fungi like candida and so on. And I just want to ask if the package has already arrived at your place. Yes. Okay. Fine. And
yes.
And what else? I think I also sent a ruthenium complex, which is a complex with Mycetoma, that fragrance, which I received recently from IFF. Finally, I must say, after some months. And yes, I think that's the current state of metal complexes. I think there's also a platinum compound with that COD ligand I sent some weeks ago already. I think these are all metal compounds in the pipeline now.
Okay, fine. Thank you very much. The next objective would be with regards to UCL. So that's my objective. I was supposed to summarize the latest in vitro and in vivo results for the compounds that we tested since July, I think. So I'll just share the screen. I think. So one thing to mention straight away is I got a bit confused about which compounds from other people got screened as well. So I would prefer to update the whole list if for example, Ma would send me the list of the compounds that have been tested, because a lot of entries have been added to the master list. So it's a bit difficult to navigate like which ones have been tested like since July, just not to miss like any of them. Would that be possible to do? Wendy, Ma, would that be possible?
Yeah, I think it will be possible. I just updated all the survival percentages as well in the master list. So all the data should be there. We have to double check.
It's just I'm not clear from which point to start. That's the issue, because it's like lots of compounds added, and then I know my compounds like from where to start, like with the scheme generation, but like others it's a bit like confusing, because a lot of Bernard's compounds have been analyzed as well.
Yeah, so, but they should have in the master list to which series they belong. So you should select series 1.
But from where?
When
we were talking with Matt, he said July, August, like the latest, like in vitro and vivo screen. Okay, I'll have a look at this again.
But all the compounds in the master list, they are linked to a certain series. So if you are only interested in the Fennery moles, you simply select series 1. What I usually do is I copy it to Excel so that I can easy select certain properties in the master sheet.
From what I understand, like Matt asked me to summarize like everything like in vitro, in vivo that has been analyzed since July this year, which means it's UCL, probably University of Sydney, if I'm not mistaken. Some compounds have been like analyzed as well, and a lot of Bernard's compounds.
Maybe what you could do is yeah, because I see the problem because you won't know like when July batch starts. But
because we have only the year screen, like 2021, 2022, but like what is the starting point? It's like
what you can do is just cross if you just select series 1 and then cross-check that with what we've already have in the manuscript, that might help. Not the perfect solution, but that might assist with that.
Okay. So at the moment, I have summarized like all the latest screening mainly from the catexymes, and there was like one less phenomenal, DM61 and 1, which was potent with the indolin potency. So this scheme essentially summarizes like all the catexymes that have been made, and the last row refers to the catexymes that have not been tested yet, and which would have to be dispatched in the near future, including the analysis involves all the in vitro and vivo data, IC50 values, and also the log D. And I found like an interesting trend. I don't know whether it's still enough. Probably we need like more molecules to decide on the trend, but what has been apparent like so far, that the trend that we found with log D with series 1 phenarmols is not exactly the same for catexymes. So all the green compounds, especially in the first row, like the first round synthesis, show that like higher log D values give like potent results. And as soon as it decreases, it gives non-potent compounds. But of course, there are other reasons. So for example, this the third row refers to compounds which did not have the pyridine ring, so there was like no coordination. So we knew that it would be a negative control. But like MycetOS 68.1 has like a log D value of less than 3. So that's significantly less than what we have like in the first rows, like for the Perifenox, which is like 3.4, or very high values for 48.1, which is like almost log D equals to 4, or very high values like almost equaling to 5, like in the case with the benzoyl ring in the R region. Yes, so at the moment the trend looks like it's that mainly hydrocarbon chains are giving the best results.
Dimitri?
Yeah.
This is also what we saw with the phenaremol. So in vitro, we see the opposite relation. So we saw in general more activity with the higher log D values, but only those which had in vitro potency with low log D values were also able to inhibit prolong larval survival. So we did see the same phenomenon in vitro, but in vivo we saw the opposite trend.
Okay. Wait, but like here I can see that it, but again, like for the catexymes, like so the first number like 3.7, for example, and 4.8, this is like 100 micromolar and then 25, and they're like similar, so that's slightly different to what we had like in phenarmols.
No, in vitro we did find relatively higher log Ds for the compounds which were able to inhibit the fungus in vitro. However, if you then look in vivo, then the compounds with the low log D were able to prolong larval survival. So of course, within the large pile of compounds which showed activity against Madurella mycetomatis, you had both high and low log D values, but in general more high log D values than low log D values, but only those compounds which did have a low log D value and were able to inhibit fungal growth in vitro, those had a higher chance to prolong larval survival in the in vivo model. So if you take the whole batch of molecules for the venery molds, we did see a trend towards higher log D in vitro, or a statistical significant difference. But if you took in vivo results, you saw the opposite. So
those that proceeded to
yeah.
the in vivo stage had the opposite trend to the one that in vitro. Okay.
Yeah.
But again, as I said, like in row 3, like with exception to 68.1, we know exactly why others are negative. It's because like they're lacking the coordination to the heme iron mainly, or there is like no proper like moiety like in the R group for the oxime. With regards to IC50 values, the potent compounds gave similar values, which is quite good, I would guess. It's in the range 0.1 to 0.4. And just as a reference, the Perfinox on the top left has a IC50 of 0.1. And the good part is like the other compounds like that are labeled green, which are potent, have like similar IC50 values. Which is a bit hard to distinguish between them, like which ones to leave out, like which ones like should we develop further. But I guess like as we expand on the library, we will see more trends. Also would like to just give a quick update on the future molecules that will be tested like in the future. So most of those that you see in the last row have been completed. So I would guess shortly we will probably send another batch of compounds. Definitely before New Year, but I think it's going to happen like much earlier before that. One of the latest things that I did like the caroxime analog with the pentane ring. But I got a unexpected result. Well, it's expected. It's just the equivalence was too high, essentially. So usually the protocol that I would follow would be the alkylation of the OH precursor, and there would be 1.5 equivalents of the substrate, which was like very high. I mean, this protocol was followed like exactly the same as in the literature and worked perfectly well for the other few caroximes, but in this case it was the iota group, which is a better living group, and the equivalence was too high, so I got a double alkylation at the end, which is not bad at the if you think about it, because we were talking in one of the previous meetings that I think Bernard mentioned this, that inhibiting the nitrogen would give a good positive control in order to prove that like coordination is quite important, like in this case, even though we had already like a few negative controls, but me and Matt thought that it would still be like, because we got like this unexpected result, it would be still good to put this compound for testing as well. And again, like I will change the I have changed the protocol, and I will do this compound again. So I would expect it to get the right thing like next time, I guess. DM71, DM72, and DM78 are ready. 76 haven't worked for a few times, but I think I got it like this week, but I'll double check. And 74, I'm still waiting for the reagent for the precursor, phthalimide precursor, to come. So there's like a slight backlog. Apart from that, like I would expect like us to send like a new batch like shortly. I guess that's all on UCL updates for this meeting on the chemistry.
Yes, thanks, Dimitri. Yes, this pyridinium compound looks quite interesting in terms of a negative control, a possible negative control. And yes, we will see. There are also examples of pyridinium compounds, analkyl pyridiniums, which have improved activities, biological activities, for instance, against certain parasites. So it will be exciting to see how far this negative control can add to
And this
the
is something,
result you're
Bernard, that you
getting.
mentioned in one of the previous meetings. You specified, like, so you said, like, it would be good to have like nitrogen blocked in order to see whether, like, it's definitely, well, proof of concept studies essentially. It was unexpected, to be honest, because the protocol worked perfectly well. But usually I would use like the chloro substrates, and this time it was like iodo. So the equivalence was too high essentially, so it got attacked from the other side, which is not bad at the end, as I said. So we'll test this and see. Yeah, I'm also curious what result it will give. So we will see. Any more questions on the synthesis part? Group members. Or shall we move on to the next part?
Yeah, sounds good.
Yeah. So the next one on the list would be chemistry. Okay, so I covered my chemistry in part 2. Han, do you have any information from Kimberly? And
Yes,
from other members of your group on the...
Yes, I think
next
Kimberly has put an update in for the molecules that have been made and our plans. So that's going to be down in the comment section there. I think that's why she ticks off that next point. So I think that one has been done. So we should be able to use that as a reference to see which one has been done. So like for any schools that wants to join our projects to make some compounds, we can probably just use that one for now. So that point has been done. Also, Clementine has updated the master list with compound from series from SSP and also from Breaking Good for 2021 and 2022. So the details are now up on the master list as well. I have put my scheme. So this like heading 2.2, I've put the schemes on the previous meeting already. So I'm currently just getting characterization data done for these for this batch. Once I'm happy with it, I'll actually ship that over to Wendy.
Can you please quickly remind which targets are we talking about?
So for myself, that will be a couple of morphin aromas.
Oh, it's the ones that you, the scheme that you showed last meeting. Yeah, I remember now.
So yeah, I've already put the schemes up on there for reference. I will haven't got time yet, but I will put those. I will make new MyOS code for those and then put it into the master list as well.
Other samples incoming and desired. So the EPIChem analysis. I have not generated the scheme because I got like flu at the last moment, but I have like everything in Data Warrior file. So I would expect this to be posted like this week, my final list, and then we can combine this with Hunk's. It's just I don't have the ChemDraw scheme, everything like in the raw file.
Yeah.
From what I remember, so there were like few compounds. There were like a lot of compounds that Hank previously proposed were with the hydroxy group and the central carbon atom for the phenaramals. So he was looking at the pyridine rings, like the additional motifs on the pyridine ring also. For that one. Okay, so it's better like for me to post my ideas and then we combine this because it's a bit like hard to talk about this without the scheme. So my apologies on that. But yeah, I'm done with the analysis. I'll just post the scheme like this week for sure.
Okay, sounds
and
good.
then we can, yeah. but I got like at least few compounds from that, like with that central OH atom that you proposed, Hang, as well. But we'll have discussion like as soon as this will be posted, I guess.
Yeah. So if anything, if we have any duplicates, and that's great. That means that we just agree that it's a high priority, and then we'll just combine those together. Yeah, just post it on this GitHub issue here, and I'll also share mine. So I'll reshare mine, and then we'll go from there.
Have you only? So I think I have it here. Yeah, this scheme that you posted. Have you only concentrated your efforts only on the scaffold with the hydroxy moiety in the central carbon atom, or were there other motives? Because I approached it slightly different. So I looked like at the generalized scheme, and when we proposed that there would be like, so for instance, it will load up in a sec. Should load up. Yeah, so for instance, you concentrated your efforts on the OH, on the central carbon atom, but I also looked at the other motives, like for example, the amine, the methoxy. Have you got like any results on that?
No, I don't think at the time when I did that search, I did not focus on the other
Okay.
ones. So I'm more.
So I guess like I'll update like more expanded version of like, because I looked at different fragments in Data Warrior, so we can either accept them or just like discard them, like if not needed. Yeah.
Yeah, that sounds good.
So what's next? Antimalarial compounds, Funk, the second one.
Yes, I've sent Rowan an email this morning. Sorry, I left it very late. I totally forgot about it. But yeah, once he's give me a reply, I will update with everyone.
So this one, I think I did assign the codes for the third objective. So I'll probably take this because we mentioned that the master list is up to date now. So I guess this is complete. Bernard, I think you mentioned about your compounds in the master list as well. So that's up to date as well. So I'll take this. Is that correct, right?
Yes. Yeah. I had some problems at the beginning because of some salt forms, which number to take.
That's exactly what I thought. Yeah, that's exactly what I thought.
But I've corrected it meanwhile. I think it's correct now.
You mean the coding system? Because I don't remember like having the codes for the metal complexes. That's something that we need to consider and update.
Yeah, that's another point. Yes. But at the beginning with the sodium salts or the chloride salts and so on, I had the problem to choose the right number for this for these compounds. But meanwhile, it's been corrected. And in terms of the middle
we still need to look at this. Okay, Matt, I don't know the status of the metal complexes that he was supposed to get. I haven't spoken to him about this, and because he's not in, I'm not sure. I will raise this before the next meeting when he's back.
I'll just shelve that point in the minutes for now, so we'll get back to Matt later once he's available.
Before we go to the paper writing, maybe it's easier if we show the biological evaluation now, the last results. Mark, could you share your screen?
Yes. But
I'll stop sharing mine. Sorry.
maybe yeah. Can you see my screen now?
Yes.
Hello. Yeah. Okay. Today is the last update of the in vivo screening of MycetOS Open Source 2022. First, we mentioned the compounds from selected from the in vitro test this summer. But today we Today we will update all data about series 1 phenomorphs. It includes 2 phenomorphs selected from last summer and 7 phenomorphs selected from this summer. Yeah. This is a summary in the table. We can see in total 9 compounds we test. The DM42 is toxic, and the DM44 has significant in larvae treatment in vivo. So this is results in figures, and we didn't separate the compounds by months, but yeah, we separated them in the series. So we update all data in by series series 1 and by Myers number. So if maybe you can see all the data easily. And this is a toxic test about the DM42.
So in this graph, you can see that the green line, if you can go back, Mark, is above the black line. So that means that there is increased survival, and it's only for Myers 310. Yeah, so we use this information to do the statistical analysis. And Mark, now you can continue.
Yeah. And all of data of molecular weight and log D, rot bonds and log P and flexibility was calculated by Ben, so 6 Ben. And we we calculate all data in the log D with survival 10 days. Visually, only a trend between the log D and the survival percentage was noted. Then we calculate, therefore we performed the main Whitney test to see if there was a correct correlation between a survival at day 10 and log D. We split at log D of 2.5. Then a significant p-value is between the log D below 2.5 and log D beyond 2.5. Then it's a small summary.
Ma, can I add to that? So since Ben did the previous statistical analysis, when we have the final set, I would like to ask Ben to do another statistical analysis, because this is a very simple one. But there probably are more sophisticated tests he can perform as well, to see if there's a statistical correlation. But if you simply ask the question, do we see enhanced fungal survival for compounds with a log D lower than 2.5 compared to compounds higher than log D 2.5, then with Mann-Whitney there is. But yeah, it's a simple test. So
I want to mention that Ben Perry will leave the NDI soon, and his successor will be Dr. Luisa Cruz, who is located in Rio de Janeiro. So I'm not sure if Ben will do anything anymore. It will be better to contact Dr. Cruz then, I suppose.
So which Dr. Cruz?
Dr. Luisa Cruz from the NDI.
Okay. Yeah. Well, he didn't mention it when I asked about the log D values.
Really?
He simply sent it to me.
He wrote me an email last week and about that matter, that he will leave soon and that Dr. Cruz will be his successor then.
Okay.
I wonder as well if we move the goalpost. So say if it's not 2.5, it is a different value. Would that make a difference for the test?
Yeah, I tried 3.0 and then there's no statistical correlation anymore.
Okay, listen.
Because I asked myself the same question and I thought, well, 3, it looks like a nice number because you see most of them on the left of the tree. but no, it only I only tested 3 and 2.5, but 2.5 gave the positive correlation and 3 didn't.
Right, makes sense.
Okay, so it's a final summary. We finished the in vivo efficacy of phenomals selected for this summer, and then calculated the remaining log D values. Then we updated the log D results. By plotting the survival percentage to the log D, a significant higher survival percentage was obtained for Madurella mycetomatis infected larvae treated with phenylalanine analogs with log D below 2.5 compared to those treated with phenylalanine analogs with log D beyond 2.5. Then we will start the compound screening next month. And yeah, we did receive the baby's compounds last Friday. Then yeah, we will start the screening campaign next month. So that's all. Any question?
Yeah, I just want to confirm. So I'm gonna put this into the minutes so we don't forget. So we're gonna try and get see if Ben or if Dr. Cruz can actually come up with a slightly or another method of lab it needs to see if there's any correlation. Is that what we're trying to do?
Yeah, so in the first publication, we didn't specify the statistical method. Ma, maybe could you stop the sound? Because I'm not sure where, but you have a lot of background noise. So he showed us some correlation between log D and survival, looking at all the molecules we tested. So not only the phenylalanines, but it was all the molecules. I'm not sure if he did perform a statistical analysis on which. So that's what I would like to confirm. If he did do a statistical analysis and if he could repeat that analysis for this data set. It's not a very large data set, and if he already done it, probably it's sorry, simply uploading the data in his program. Because I can imagine that there is a statistical analysis you can be done, not splitting at 2 values, but taking the whole into consideration. Because you do see a trend that a higher percentage survival on the left side of the graph and lower on the right side of the graph. But I just simply made a cut 2.5 one way, 2.5 the other way. But of course you can do statistical analysis taking all the values into consideration. But I'm not a statistical expert, so I thought maybe he had better statistical ways of looking at this, because they probably did it much more often than I did.
Yeah. So can I just leave that with you to figure out a bit further?
Yes, make it an action point for me, and then I will contact Ben. And if Ben doesn't want to do it, he will probably direct me towards his colleague, as Bernard mentioned.
Yeah. All right. Thanks.
But of course, it could be that he's still willing to do this because this is really the final part of the manuscript, and then it's done.
So I guess we probably will move to the next objective, which is paper writing. So just before we continue with this, on behalf of UCL part, I would like to say that I added all the experimental data. I cleared all the comments that were, most of them, with regards to proper writing style, bolding, and other stuff, extra stuff that should have been removed from the experimental section. This is all clear now in both in vivo sections of the paper and in vitro as well. So everything is there so far. So I think
Yeah.
Peter and Alice reviewed the paper several times before, and I can see that this objective that was assigned to Hank has been completed as well. I remember talking to Matt, and he said like as soon as we have like a more updated version, he will go through this again.
So
Yeah, I
yours-
went to it this week actually, or last week.
Yeah, I saw.
I updated the in vivo data. So the new graphs which I showed now are also in the manuscript. I only had to double check the in vitro counts. So we stated now for the in vitro susceptibility testing that we tested 185 compounds. But since writing that section, this summer happened. So probably I need to simple count how many compounds we did screen in the end for the Fennery moles and how many positive hits, etc. So I need to update that section, but I didn't came around to it yet. I did the in vivo part first, because then I could analyze everything which we just showed.
Okay.
Peter just got back to me today because he needed some extra time to finish that. So the version 5 is the one that Peter has reviewed. Is it possible if you have done any, has any other updates been put into version 4 before Peter got back to me? If it's yes, is it possible if you can port that over to the latest version, like version 5?
So I worked in the version which was on the
Number 4.
Yeah, so there I updated everything
me.
on the biology.
I actually noticed that version 5 appeared only today because I was looking at the version 4 only yesterday. So I just saw that the version 5 appeared today before the meeting.
Yeah, and I worked in version 4 because you mentioned last time, Hang, that that he would only go. He asked Matt and me to go over it first, and then he would go back to the manuscript and look at it because he didn't have time. So that's when I assumed that he was not working on it, and version 4 was the most up-to-date version.
It was until today. How exactly was it done actually? How did like version 5 appear? Like was it like superseded like from version 4? You just renamed the file, or you created like a new one?
Okay, so Peter managed to get the so Peter actually asked for some extra time, but then it actually took him a bit longer. I actually took down that comments because Peter just asked for a few more days. So he's worked on it, but I'm not sure if he works on the. He got back to me late yesterday, actually. So like at I think 11 p.m. yesterday. So I think if yours has been in there before that, he might have actually got reviewed on the new.
Dimitri, for me, it's quite easy to see what I changed. Is it? Do you also have a spot where it's quite easy to see, because then we can simply open version 5 and see?
I mean, it's not a problem because I can see like that it's updating with the latest version. So I just opened the up-to-date file. So for example, the Veneramos 5 appeared on its own, like so it's updating everything as it is, like in the Dropbox. So it's synchronizing like in the right way. So I'm pretty sure like there's no duplication or like addition to the wrong file.
Is it possible if Wendy and Dimitri just double check that version 5 also has your update as well? If that's possible. I'm sorry for the slide.
Yeah, I checked. I looked at it quickly. So it's all the things that I added recently prior to our October meeting. So it's up to date, actually. Okay. Yeah, on my part.
If the one you opened just now was displaying the figures you showed, then it's also with my comments, because I updated. Yeah, is this showing panel? Yeah, this is with my comments as well.
Okay. Okay.
Yeah. And I guess, yeah. Hank, what's the status of your experimental section results? You still need to do like finish formatting, like the bolding, fixing, like how?
Yes, I will get to that. So Peter's mentioned that there's also some comments addressed to me as well. If you can leave this draft with me to the end of this week, I will address the comments that was left for me as well. I think he left some, and he also mentioned that Matt has some. So I'll go and I'll go over whatever was addressed to me, and then I'll update that. Just give me to the end of the week.
Yeah. Yeah. So on my part, like, yeah, it's all the JCF coupling values. Yeah, everything is up here, and it's been formatted in the right way as been asked. So I guess like in terms of like my experimental, I'm fully done with this. But I noticed, I'm pretty sure that I cleared all the controversial like formatting issues yesterday. I double checked this yesterday, but for some reason, like I noticed like now in version 5, some things disappear again. I'm not sure why this is happening. But I'll go through this again. I'm not sure.
Yeah, I think this might just be something that we just have to be diligent. So it might pop up again. It's just that different people.
I checked. I'm telling you, I checked everything
as in
before
like
the meeting. So
between like some people use Windows, some people use Mac. So that might actually cause formatting. It's actually might not actually be your fault at all. Like it's just a software thing. So we just might just have to, like, if I say something, I'll just fix it.
Yeah, and probably
bolding was cleaned as well. So yeah, I'll go through this as well. But it was clean in version 4, in Finarmos 4, and in 5. But I will go through this. It's not a big deal. There are not many things that are not right, I guess. So there shouldn't be a big issue with this. Sorry,
yeah,
Wendy.
and probably when we're almost close to submission, then we need to take it out of Dropbox and put it in Word of the computer, probably of Matt or Dimitri, as Matt was going to be the senior author. And they have to, that the one who's going to submit simply needs to go over it one more time to change these things, because indeed it will probably be that someone use Mac, other one I use a normal computer, so Windows computer.
I use Mac.
Yeah, so yeah, that's the downside. But unfortunately, the other program, the shared doc program, didn't work for the figures. So yeah, it was one evil or the other evil.
Yeah,
I mean, it works pretty well for the MacBook Pro. So for the Word in MacBook, I get like I just save the file and then synchronizes straight into the Dropbox. But sometimes it might get like some data files in the wrong format. That's the only issue. But otherwise, it's more or less.
It's more has to do with the coding of the fonts. Like it's gonna take too much effort to figure out what's wrong with it. So like I think the most efficient way that we do is we'll try to fix whatever we see, but like towards the end, we'll just have one person to go through that and make that final, make sure that it's unified. But otherwise, yeah, it's the coding of the font, but and then like each different system like Windows or Linux or Mac, they might have a.
With regards to final review, all Matt asked me about, and we talked about this like several times before. He wants essentially as complete version as possible, so that he goes through it like without like any major additions like in the future. So that's what he's been asking. But I guess like because Hank has added like all of his stuff as well, and all he needs to do is like format a bit more, like fix the bolding and stuff like this, just like fix like few comments from Kimberly, and then we're ready to go like with asking Matt to review the more or less like final version, I guess. Right? Yeah.
Yes, I've also have assembled my spectra as well for all of my compounds. So it is now also up on Dropbox in a
Yeah, I saw the updated file just before the meeting, the NMR compilation. It's PowerPoint, right? Format at the moment.
But I'm happy. I still have the like all of the images. So if we decided to go into a I don't know Word document or something like that, I can just put it into a different form. But it's currently in PowerPoint.
Wendy, where should those files be added? Like, should they still be like in the Word format or in the PowerPoint? Because I'm yet to add mine.
I'm not sure because I'm not familiar, because I thought we are going to do this to a chemistry journal.
Yeah.
Depends on what the journal asks for the supplementary files, I guess. So maybe you can check the journal
I think
site.
it's still going to be the Word format because supporting information, like everything essentially in the research papers are in PDF files usually, but you submit everything in the Word file, so it has to be.
Yeah, because if it's PDF, and then we can still just convert that PowerPoint into a PDF. But otherwise, I just leave it there for now. And if we need to do it later, we can change it back to a different format.
I will speak to Matt about this, and we'll probably start.
Because every journal has its own rules. So
yeah,
for some journals, it really doesn't. Yeah, it doesn't matter. You can even upload PowerPoints or whatever you want, but for other journals, it does.
Yeah, it's there for now. It's just for reference. So we don't we just have it.
And coming to the final part, the admin stuff, most of this has been ticked. So I think it's mostly things that had to be done before the meeting. Things like generating Zoom link. Wendy mentioned at the beginning of our meeting that like the Teams and Zoom links have been fixed. So I don't think the invites are sorted as well, as far as I know. I saw it like in the invitation links in the future for the future meetings. At least it's sorted like in my Outlook calendar, so there are no problems. Breaking good compounds, smiles. I saw Kimberly and Hank updating this on the master list, so that's sorted as well, as I mentioned before. A presentation has been submitted like at the end of last meeting, so that's sorted as well. So I guess like we covered everything for today, unless anyone else has any suggestions.
We've actually ticked a lot of points in this meeting, so that looks awesome.
Yeah. So I guess unless anyone has any other things to cover, we can finish off this meeting. Anything else?
No, I think Hank took the minutes now. You were convening so that what was the rotation like? So the one who takes minutes has the time off next time.
Yeah.
That means that Dimitri is taking the or UCL is taking the minutes next time. I'm convening, and that's the rotation, isn't it?
And Hunga's chilling next time.
Is that how it is? So it's gonna be Erasmus to what? Sorry, I lost. Could you repeat?
So in the next meeting, I guess Erasmus will be leading the meeting. UCL will be taking minutes, and you'll just be giving updates, I guess.
Okay, sounds good.
I will update this table here now, just after we finish straight away, not to forget. Okay. All good then. Thank you very much for joining us today.
See you all next time.
Yeah, right. See you in November.
Enjoy your day/night. I think most of it's our day, but I'm going to enjoy my evening.
Good night, Hank.
All right. See everyone.
Goodbye. Bye, Bernard.
Bye.
Bye, Wendy.
Bye-bye.
Bye, Mom.